My name is Walter Jayasinghe, the Director of Cultural Affairs at the Sri Lanka Foundation and the Director of Operations at SLF Academy of Performing Arts. On behalf of the Sri Lanka Foundation, I'd like to welcome you to our third medical webinar on COVID-19, featuring our very own doctors from California, Minnesota, and Florida. Before we begin, I'd like to go over a few things with you. First, a few rules. Sri Lanka Foundation requests all attendees to follow the house rules for the webinar. Mute your audio if lines are open. Keep your phone muted whenever you are not speaking. This helps avoid unwanted background noises that can distract other participants. Sri Lanka Foundation will be recording this webinar. As such, we ask the attendees not to record any conversations. Be respectful. Keep your comments helpful and considerate of the host and other participants. During the questions and answer session, you may ask the panel of doctors any questions you may have through the chat line seen at the bottom of your screen. The moderator will direct the questions to the appropriate speaker. Disclaimer. This webinar is designated to provide helpful advice and information on the subject of the novel coronavirus, also called COVID-19. Individual diagnosis and concerns should be directed to each participant's personal physician. The authors and hosting company are not responsible for any specific condition that may require medical treatment and supervision and are not liable for any damages or negative consequences arising from this webinar. Thank you. And now let me hand over the webinar platform to our host, Keshini Vijayagunratne, the Director of Project Management and Public Relations at the Sri Lanka Foundation, and our co-host, Chamu Damarasinghe, the Media Director at the Sri Lanka Foundation, who will be handling the tech work today. Over to you, Keshini.
Thank you, Archula, for covering that important information. You always do such a great job. Thank you for welcoming the people. On behalf of the entire Sri Lanka Foundation, I would once again like to welcome and thank all of the attendees, the distinguished guests, the Sri Lanka Foundation staff, and the panel of doctors, the Academy of Performing Arts staff, and the teachers and the DG Studio staff for joining us on our third medical webinar. We are so proud, right, the Sri Lanka Foundation staff, aren't we?
Yes. Yes, we are.
I have to start off by appreciating Dr. Walter Jayasinghe. I don't have time to talk about Dr. Walter Jayasinghe, but in little words, it is his vision to keep the communities connected and educated, to bring awareness, and especially now with the pandemic still spreading. I also want to thank Dr. Dishan Jayasinghe, our president. Thank you, Dishan, for all your support. And Aisha Jayasinghe, the vice president. Thank you for your support as well. Shirani, our chief operating officer, Achala, director of cultural affairs, Chamod, the media director, Chamod, for all your support. I mean, he's such a big support. He has been working day and night to put these webinars together. And the doctors, you know that's true, right? The Academy of Performing Arts, Lal Tilakaratne, thank you for your support. And DG Studio, Zupli, for all your support as well, the creative director. I also have to thank some community organization people that have been with me the last couple of months. Rakshita Disanaika, the Ananda College president, thank you for all your support. Sri Lanka Youth Organization, Aruni Ganewata. Good News Jayam, you talk to me day and night about the webinars. Thank you for all your support. And last but not least, Nandi Jasuntulianer. My gosh, you are like my brother, Nandi. I talk to you day and night. Thank you for all your support. I also want to thank the panel of doctors for dedicating their time and support. Very especially our director of the Sri Lanka Foundation, Medical Dr. Deepti Jayasekara. I couldn't have done this without you, Dr. Deepti, honestly. Thank you so much. Without further delay, it is my pleasure, and I'm so proud to invite Dr. Walter Jayasinghe, the founder of the Sri Lanka Foundation, to speak. Dr. Walter, is all yours.
Good morning, afternoon, evening to everybody. First and foremost, I thank all the people that Keshini thanked. In addition, special thanks to Dr. Deepti Jayasekara, our medical director, putting the panelists together. And Dr. Anil Jayasinghe, thank you very much for consenting to be on this webinar. Thank you very much, and thank you everybody enjoyed.
Now I'm honored to invite our Ambassador, His Excellency Rodney PERAHERA. Ambassador, you're online, right?
Let me start with Ayubowan. May all of you have long lives. So thank you, Sri Lanka Foundation, once again for this informative platform to hear our very own accomplished medical professionals from different parts of the US, Dr. Deepthi, Dr. Malini, Dr. Devinda, Dr. Lushanta. Thank you very much for devoting your time on a Sunday. We are also privileged to have two eminent practitioners from Sri Lanka, Dr. Walter Jayasinghe, I think a mentor to some of our panelists, and Dr. Jayasinghe, a key contributor to the COVID-19 mitigation success in Sri Lanka. We are proud that the excellent education system in Sri Lanka has produced so many top-notch persons. We salute your professional achievements and your service to respective countries. This event, like others before, is possible only because of the selfless dedication of Dr. Walter Jayasinghe, whose life's achievements continue to inspire many others. I thank Dr. Walter J., the best example of Sri Lanka's education system in the US. Aisha, Dishan, the Sri Lankan Foundation, dynamic team, Keshini, Shirani, Achala, Chamod, for continuing to serve the wider Sri Lankan community. I strongly believe the Sri Lanka Foundation can play a critical role in bringing Sri Lanka and the US together even more closer, as witnessed today, as the foundation can provide the bridge to fruitful cooperation by harnessing professional expertise for many to benefit from. Similarly, I'm certain we can extend this type of activity to forge closer links in promoting the wonderful Academy of Performing Arts to take our proud culture to all corners of the world. So today, let's have a successful and informative session. Thank you all very much.
Thank you, Ambassador Naibowan. I'm so proud to present to you our next guest. He's going to inspire everyone when he shares his story, how he contained the novel coronavirus, COVID-19 in Sri Lanka. Is none other than the Director General. And now he is the Secretary to the Ministry of Environment. None other than, here we go, Dr. Anil Jayasinghe, lawyers.
Thank you, Keshini. It's a privilege for me to speak to Sri Lankans in the US, and I thank Sri Lankan Foundation for inviting me to speak. So I believe that I have to talk on the reasons for success in COVID battle in Sri Lanka. So without further ado, I would like to take one by one the reasons as I perceive. Now actually the Sri Lanka, the COVID was limited to clusters, and we never had social transmission. So the first reason why we were successful was the strong public health system in Sri Lanka. This is a time-tested public health system and the public health indicators, and during 30-year war. So therefore, I think we must dedicate the strong public health system as the main reason for Sri Lanka's success. And then, of course, strong political leadership. I attribute to His Excellency the President. I believe his experience in battlefield also would have helped him, and his no-nonsense approach and decision-making, kind of apolitical nature in directing this battle and facilitation. He facilitated us. So that's a very strong point in Sri Lanka, the strong political leadership. Of course, then the strong technical leadership. Now, when it comes to technical leadership, creating technical teams, technical direction, and the not getting confused, and also not confusing others. So these were very important. We never got panic. So that the strong technical leadership certainly paved the way for the success so far. And then putting simple epidemiology into practice and not taking any risks. I think the epidemiology is a historical science, and there are fundamentals in epidemiology. So I believe in Sri Lanka, we practice the fundamentals correctly. That's a major reason. Now, when it comes to contact tracing, we were very successful, and we quarantine into practice. We put quarantine into practice in a strong manner. And then I think one key decision which paved the way for Sri Lanka was admitting all COVID-19 positive patients. You know, we never allowed any COVID-19 positive patient to be at home. Of course, you know, countries practice in various ways, but you know, these things allowed us to control efficiently and effectively this disease. And another reason is starting this battle very early, efficiently and effectively. Now, it was on 23rd of January that we established National Action Committee Against COVID-19. It was on 25th of January that we established screening facilities at ports of entries. And following which I went on a visit to Geneva, and I didn't see in any of these airports those facilities. And then it was on 25th January that we established COVID PCR testing facilities at MRI. And I have to mention you that it was only in 11th March that WHO declared COVID-19 a pandemic. So therefore, in January itself, we started the battle. And then another main reason is using core strengths of various organizations. Now, it was not only health services. Now, though the health ministry, the health services were leading, but then His Excellency, the government, the health ministry, we all realized that certain core strengths are with others. Just to mention quarantine. Now, when we wanted to quarantine so many thousands, hundreds, sometimes 200, 300 per day, of course, health ministry alone couldn't have done that. So therefore, the Sri Lanka Army came into being. Of course, they worked heavily, and then of course supported by the Air Force and the Navy as well. And when it comes to contact tracing, in basic epidemiology, we do contact tracing by ourselves in local spread of certain diseases. But when it comes to COVID-19, contact tracing is not easy. So therefore, we got the help of SIS or the intelligence services, using by the way of their technology. They have the best of technology in Sri Lanka when it comes to this kind of work. And the tool, that's the main thing, the tool that we used, that was Quarantine and Disease Prevention Ordinance, enacted in 1897. So this was actually enacted during British period, and all the powers vested with the Director General of Health Services under this quarantine basically were used for this battle. Now, the entire country was declared as a disease entity earlier on, and then all quarantine centers were commissioned, taken over. using the ordinance. And then the process of quarantine was conducted once again using the powers vested with DGHS under this ordinance, and then locking down areas and then contact tracing. So therefore, now what I mentioned, all these were able to function due to this tool, which is the ordinance. And then of course there are other reasons like managing media. We could manage the conventional and the social media both in a manner that they were in support of the cause. So earlier on there were some media went on wrong path, but then we were able to get the act together. And then the rising to the occasion by the health staff. If not for the health staff, we would have once again failed. And of course, public cooperation. Without public cooperation, we cannot do anything. And ultimately, I perceive that the country, the authorities, the public, everybody got this feeling of made in Sri Lanka solution. It was made in Sri Lanka. So therefore, I think this success is owned by entire public of Sri Lanka. Thank you.
Thank you, Dr. Anil. That is amazing. Thank you for sharing. Thank you for sharing that story. Wow, it really inspires everyone. Our next guest actually is very dear to some of our local doctors in California that are very successful today. Actually, back in the days, he was their professor, their teacher. Right now, actually, currently, he's the chairperson for the University of Colombo Medical Faculty and former Department of Medical Head of the Humanities and Faculty of Medicine, Dr. Walter Jayasinghe.
Thank you very much, Kesini. Can you hear me?
Yes.
Yeah, and thank you very much to the foundation, and it's lovely to be with you once again. Just to reiterate what my friend Dr. Anil Jayasinghe said, I think they've done. I mean, Anil was leading the whole battle, and I think we've been very, very fortunate that our rates are so low because of the actions taken by the government and the Ministry of Health and the security forces. And as he mentioned, it was very decisive early action taken in several sectors and coordinated by the government. And I would like to just touch on some of the other areas. For example, the numbers which we have got. So far, we have had 3,283 cases detected. And as Anil mentioned, all these patients were handled or managed in the hospitals. There were hospitals which were designated at one stage. There were 12 hospitals, and all positive PCR, even from the quarantine centers or detected from any screening, was sent to these hospitals. So we had people confined in institutions, and they were tested at least on 2 or 3 occasions before being discharged. And the types of patients we've had, so far we've had only 13 deaths, which is good. I mean, that's a fairly low case fatality rate compared to countries such as Vietnam. Now, Vietnam is considered an example because of its very low rates, but their case fatality rate is much higher than Sri Lanka. Now, of the cases who have gone into hospital, there are 1 or 2 publications which have come out, and I must say that the College of Community Physicians of Sri Lanka have just come out with a very useful volume on COVID in Sri Lanka. You can access it. Access is free, and it's an open access journal. And the interim analysis of 100 cases has been published by the physicians working in the infectious diseases hospital. And so far, we've only had 13 deaths. And the deaths are predominantly in people who have comorbidities, such as diabetes, hypertension, cancers, and stroke. We've had probably 2 who didn't have many comorbidities and died of the severe respiratory syndrome associated with COVID. So all in all, if you look at the symptomatology, it's standard what we see elsewhere. And the difference we see is that in people who are coming from the quarantine centers, most of them are asymptomatic. Probably about 77% of them are asymptomatic. Whereas people who are found with symptoms and referred from hospitals, etc., even in that group, you get about 25% who are asymptomatic, but subsequently some of them develop symptoms. And we see the classical symptoms described in the literature. And the treatment-wise, initially we gave hydroxychloroquine, but now for asymptomatic, we are not giving other than vitamins. We don't give hydroxychloroquine. If they're symptomatic, they're given hydroxychloroquine. If they have respiratory features, then they're given enoxaparin and given oxygen and nursed in a prone position. We haven't had to use dexamethasone as such because the reports of its efficacy came only quite recently. So that's just an overview of the situation in Sri Lanka. Thank you very much once again.
Thank you, Dr. Walter Jayasinghe. Now I'm very honored to invite the president of the Sri Lanka Foundation, Dr. Jayasinghe.
Hello, everyone. Thank you all for coming. I wanted to thank everyone for coming and all the people involved. There's a lot of professionals that we have here that are sharing with you great news and understanding of the important parts of how to handle this disease. We can all be very, very, very proud of our country for handling COVID as it has and continues to do so. The unique part about, I think, our medical webinar is the difference between having a lot of false data put in your face, data that's not true and doesn't work, versus data that's actually being used and effective. That's why we put this on. Dr. J and our whole family wants to do this because there's a lot of false information. And then there's just a lot of information in general. So how do you decipher what's important and what to pay attention to in this sea of all this information? And I think that the selection Dr. Jayasinghe has put together of panelists can help us decipher this, and they show us what's actually working. And through time, we've noticed that we get more and more information that is, I believe, very helpful, and with a top level understanding that gives ease of actually applying this information. So I hope everyone pays attention to the important parts and continues to keep safe. Thank you very much.
Thank you, Dr. Deshawn. Now I would like to invite someone that I work very closely day and night, our Chief Operating Officer, Shirani Stanislaus, to introduce the panel of doctors. This is the moment you've been waiting for. Go ahead, Shirani.
Thank you, Keshini. Good morning, good afternoon, and good evening for those of you who are listening from Sri Lanka. My name is Shirani Stanislaus, the Chief Operating Officer of Sri Lanka Foundation. Today, I have the honor of introducing a panel of 4 eminent Sri Lankan physicians. First, Dr. Deepthi Jayasinghe, our moderator. Those of you who participated in our previous webinars would obviously remember him. Dr. Jayasinghe is an infectious disease specialist and clinical professor at Western University, Chief of Infectious Disease at eminent health and San Dimas hospitals, dealing with a broad array of infectious diseases. He is primarily specialized in infectious disease for over 28 years. Our next speaker is Professor Dushanta Jayasinghe. He is a professor in clinical medicine at the University of Miami. Currently, he oversees the infectious disease clinics at University of Miami Miller School of Medicine and Jackson Health System, where over 3,200 HIV-infected patients are managed. He has been a PI and co-PI, I believe it stands for Principal Investigator, in many NIH-sponsored studies in infectious diseases, drug abuse, and the site PI of many industry-sponsored infectious disease studies. He is also a member of the Center for AIDS Research, as well as a senior investigator at the Clinical and Translational Science Award, both NIH funded. He has also been a site PI in over 80 clinical trials and published extensively on HIV and hepatitis C. He was the Executive Dean for Research and Senior Associate Dean for Research at the University of Miami Miller School of Medicine. He is one of the lead investigators in the Miami Clinical and Translational Science Institute. Our next speaker will be Dr. Malini De Silva, MD, MPH. Dr. Malini De Silva is a research associate at Health Partners Institute since 2019, working on the vaccine safety data link, effectiveness of maternal Tdap administration on infant pertussis disease. I believe it is whooping cough, doctor. Team-based and technology-driven adherence intervention to improve chronic disease outcomes, hotspotting cardiometabolic disparities for simulated advances in population care, and the Center of Excellence, a network for training and epidemiology in refugee health. She is also the staff physician at Health Partners, Department of Infectious Diseases, and Travel and Tropical Medicine Center at St. Paul Ramsey County Tuberculosis Clinic. She has had her education and training as MD at Mayo Medical School, Rochester, Minnesota, and MPH at University of Minnesota School of Public Health, Minneapolis, and a certificate of tropical medicine at University of Minnesota, Minneapolis. Our final speaker is Dr. Devendra Kurera. Dr. Devendra Kurera is an emergency physician who practices in Montclair, Chino, and San Dimas, California. He earned his bachelor's degree in biology and political science from the University of Southern California in 1999. Dr. Kurera attended the Keck School of Medicine of USC on a Naval Health Science Professional Scholarship and graduated in 2003. He subsequently served in the Navy for 10 years, completing his residency at the Naval Medical Center, San Diego, and was deployed twice to the Middle East. Thank you all, and special thanks to our panel of doctors. Enjoy the very useful presentation. Now, I hand the webinar platform over to our moderator, Dr. Deepti Jayasinghe.
Hi, good morning to everybody here. Good afternoon and good evening to everybody else globally. I'm so honored to be here. Thank you once again for giving me this opportunity to speak and share my experience as an infectious disease specialist in the United States. I practice in California as a clinical professor and a specialist in infectious diseases. And I'm honored to have Prof. Walter Jayasinghe, who was one of my teachers, professors at University of Colombo, Sri Lanka. Thank you for being here, Saroj, and also hats off to Anil, who've done a tremendous job, showing tremendous amount of dedication to mitigate COVID-19 in Sri Lanka. So this is the story of COVID-19. This is the story of all the coronavirus pandemics so far. SARS 2002, MERS 2012, and COVID-19 in 2019 and 2020, and beyond. Now, when we talk about numbers or worldwide epidemiology, we have reached a somber milestone of 30 million cases worldwide, heavily underreported, but I think that's a big number. And they have reported close to 1 million deaths worldwide. Leading the way is our country, the United States of America, unfortunately, 6 million cases reported so far, and followed closely by India, Brazil, Russia, and the rest of the countries. But as we go on, you know that we have had surges in certain parts of the world. Right now, there's a surge going on in Europe, especially in England. England is close to another lockdown, we were told. France, there's another surge. Israel, there's another surge going on. And Melbourne in Australia, and so on and so forth. So this is going to continue to happen next few months, maybe a couple of years. But that is sad news. But the good news is we know exactly what the protocols are. The good news is depicted by the linear curve on this slide. The deaths, mortality has gone down after April of 2020 to a plateau that continues to go down. So now the reason why the mortality or the death rates have gone down is because of testing protocols and treatment and therapeutic protocols effectively. Then we're going to talk about US experience with COVID-19. As we know, most of the states have contained COVID-19 as we speak. The word is contained and mitigated, not controlled completely. Unfortunately, right now as we speak, there are some states such as North Dakota, South Dakota, who are experiencing high numbers once again, Midwest and South. So it's going to continue to happen. So this is not completely unexpected, but as long as we know what to do in order to return to normalcy, I think we're going to be all right. I have to reiterate the fact that we have discussed this many, many times before. When you talk about return to normalcy, you have to talk about the mitigation bundles, mitigation steps that we're going to take, right? Hand hygiene, that is hand washing and using hand sanitizers. Social distancing. The word social distancing is an oxymoron, but that is exactly what we need. Social distancing, meaning you're supposed to keep at least 6 feet apart from people, from two different people, and also not to congregate in places where things can get very ugly. Universal masking is a good thing, but so far it's been so politicized. But unfortunately, time and time again, the studies have shown that masks or the facial covers have helped control the morbidity and mortality of this illness and simply the numbers of this illness. The classic example was, Taiwan did not do any lockdowns, but they made universal masking mandatory to everybody. So in order to mitigate the virus in the future, as long as we follow these guidelines, I think we're going to be all right. We have certain sanitation guidelines in place in hospitals and workplaces such as self-screening such as screening with temperature and monitoring for symptoms. Now we can do it in the form of self-screening as well. But the most important aspects that we have going forward would be change of culture, not taking these things too easy, not letting your guard down. So change of culture, such as the holidays, certain holidays, certain behavior patterns are associated with high numbers. We know this. The numbers have shown this. So as long as you keep your life as simple as possible next few months to maybe a couple of years, avoid unnecessary congregations, avoid unnecessary big parties, or all kinds of gatherings, I think we're going to be all right. Other most important thing would be vaccines. So we have two panelists who are going to talk about vaccines. But I have to tell you, among the vaccines, we are always talking about COVID-19 vaccines, right? There are three frontliners among the COVID-19s. It's a race to success among these three to six frontliners, I should say. But equally important is influenza vaccine. In America and in Europe, we are heading into winter. So when there's winter, when there's cold weather, obviously your influenza rates are going to go up. We do not want a twindemic at this situation. Although I have the twindemic means influenza pandemic and COVID-19 pandemic. That's the last thing that you would do. So please, this is the time to go get your influenza flu shot, influenza vaccine, right? Those vaccines have been historically shown effectiveness, not 100%, but there's some partial effectivity. At its best, the true numbers are about 50 to 60%, but there are some mismatch in certain years, such as last year, right? We look at the Southern Hemisphere numbers in order to, mutations in order to formulate our own strains that we'll cover with the influenza vaccine. So we don't know what this year holds for us, but we are very confident that we can control the influenza outbreak this year around, thanks to all the mitigation steps that we are already taking. So, a pneumococcal vaccine, pneumonia vaccine is equally important. If you're 65 and above, please go to your primary care doctor and ask for your pneumonia vaccine if you haven't taken it already. There are two vaccines that we give. One is a conjugated vaccine, and the other one is a routine vaccine that we give to cover 23 variants of pneumonia, pneumococcal pneumonia. All these things are important to keep you healthy and get you through this period of COVID-19. Now, at the hospitals and workplaces, we have taken all these precautions. As you know, I don't want to spend time on these things. The signage, the waiting rooms, and other successful strategies are being implemented, especially when it comes to hospitals. I have to tell you this, please don't postpone your important screening protocols or elective procedures. Now we have taken all the steps to bring life back to normalcy in most of the hospitals, radiological labs, and other labs, right? So we have actually, we are executing plans to bring normalcy by strictly following the sanitation guidelines. Those are hardwired into our protocol. So please trust the doctors and the nurses, and then your primary care doctors and your cancer screening protocols. Please don't postpone those things. Hospitals are sometimes safer than the other workplaces or your other environments. I'm not going to talk too much about the clinical pictures of COVID-19, clinical phases or stages, the viremia stage, lung stage when we become hypoxic, and then last but not least, hyperinflammation phase, where your immune system is in overdrive, somewhat like dengue hemorrhagic shock syndrome situation. Your own immune system acts against your own body. So we've talked enough about these clinical stages. At the last meeting, I talked a lot about Kawasaki-like syndromes. About 4 to 6 weeks after certain kids get COVID-19, they're going to, we have reports of Kawasaki-like syndrome, severe joint pains, fatigue, shortness of breath, mostly heart complications. This is what we call multi-system inflammatory syndrome in children, or MIS-C. And this has been documented and bibliographed very clearly. And other COVID-19 post-COVID syndromes have been described as well, such as severe fatigue syndrome, chronic fatigue syndrome, that this could go on from 3 to 6 to 12 months, unfortunately, in certain patients, not everybody. So be prepared discuss with your primary care doctor about what to do. We have certain things that we could do in order to mitigate post-COVID fatigue syndrome. It's not just you, it happens to a lot of people. And there's a study that was done recently that shows 20% of young people who are unable to get back to their normal jobs because of severe fatigue. So you're not alone. This keeps happening. Those who have gotten COVID-19, 80% of us do very well after the 7th day, 10th day. We're going to discontinue the quarantine. CDC studies have shown that. But about 20%, so the rule is 80-20, if you call it, 20% of us will develop lung stage, pulmonary stage, where we become hypoxic. That's why it's very important to carry your, buy a pulse ox for your finger, where you can measure the oxygen saturation. Talking about other post-COVID syndromes, ventricular arrhythmias caused by and viral myocarditis, it has been described among young athletes. Now, the sports are back in action, as we are all excited about, right? NBA is there now. Football, American football, and baseball, everything is there now. You can watch those things live on TV, not live, on TV. But we have found out that certain young athletes who developed viral myocarditis, their CRP, their troponins go up, right? Now, this happened after a asymptomatic COVID period. So be mindful about these things. A lot of them do very well, but if you continue to work through post-COVID viral myocarditis, you can potentially get in trouble. Thromboembolic diseases such as deep vein thrombosis, pulmonary embolism, strokes, and heart attacks have been described. Post-viral pneumonia, I see this all the time among my patients in the ICUs. Once the patient is intubated, they can run into critical pneumonias caused by bacteria, such bacteria like gram-negative. Some of them are multi-drug resistant gram-negative bacteria because of the over-usage of antibiotics. So we have seen a lot of multi-drug resistant Pseudomonas, Morganella, Acinetobacter, Stenotrophomonas, that kind of bacteria causing post-viral pneumonia, very difficult to treat. Some of them are unfortunately carbapenem resistant, so there aren't very many antibiotics that we can give for those situations. We do use colistin and polymyxin B, these are old antibiotics, and then we have a whole bunch of new antibiotics for the carbapenem resistance. Staph aureus have been historically described as a post-viral pneumonia, like 1918, when patients died of second, third wave after the Spanish flu, they actually died of Staph aureus pneumonia. Different kinds of rashes have been described among post-COVID syndromes. Now, switching gears, I'm going to jump right into the treatment, testing protocol. So the gold standard of COVID-19 is RT-PCR, nasopharyngeal swab, right? We go one step ahead in the hospitals. We do bronchoscopies, lower respiratory PCRs have been done. Now that will remain the gold standard. Some of the gold standards are rapid now. Some of the PCRs are rapid. Now we can get the test results within 1 hour, right? We have been doing this for the past 2, 3, 4 months. ID now by Abbott, Gene Expert by Cepheid, and then BioFire test. These are among few which we use in our hospitals as point of care tests. Within 1 to 2 hours, we get the results and we start the treatment protocols right away. Unlike in other countries, we have a lot of patients who are in pulmonary stages and who are intubated. We try not to intubate them, but they end up in intubation anyway. So these have to be started right away. In order to start the COVID-19 protocols, we need to know the diagnosis. Without a confirmation, you don't want to give a toxic medication to these patients. So that's why it's very important to do these studies, RT-PCR, with a rapid diagnostic test such as ID now by Abbott, Gene Expert, or BioFire test. New diagnostic tests. I'm excited to announce saliva as a form of diagnostic test. Saliva Direct by Yale University. And right now it's been implemented in so many hospitals. Right now, as we speak, NBA national basketball season's going on with playoffs. The area that they are, they're residing is called bubble, NBA bubble, and that bubble on daily basis, those people have to go through Saliva Direct in order to diagnose COVID-19. So far, knock on wood, we haven't caught anybody, any basketball player, NBA basketball player with COVID-19 thanks to this study. and they are taking all precautions, safeguards are in place, and they are observing those things. This particular study is called SWISH study. So those are simple saliva tests. We don't need reagents for this. You just spit into a cup and then hand it over to the lab. We're going to do that at home as well pretty soon. It's been considered for FDA approval. Binax now is an Abbott test that the White House invested in. It's a $5 15-minute test, easy to use, but that's not a PCR test. It's a rapid antigen test. Now, certain hospitals, like my hospitals, we do have rapid antigens called Sophia test. So Binax now, from Abbott and Sophia tests, are very good tests to make a quick diagnosis, and they are very inexpensive, cheap. Right? So why is it superior and inferior than the PCR? Unfortunately, there are some false negatives, and false negatives about 80 to 90%, but that's okay when you have a patient where there's a high clinical suspicion for COVID-19. Then we're going to do that. If that test is negative, then we're going to do the PCR test. But if your clinical suspicion is low, then you have a negative test for rapid antigen, we don't have to do any PCR test. And in a mass scale testing, this is a great thing. And we call this two-tier testing. Rapid antigen, rapid test, within 15 minutes, we get the results, followed by PCR. CDC recommendations have changed in certain areas. Antibody testing, as we know, we don't use antibody testing, IgG and IgM testing for diagnostic purposes, but certain limited situations we do use that, such as where PCR tests fail, not one, many PCR tests fail to show positive results. If your clinical suspicion is still high for that particular patient, we can still use IgM as a diagnostic test. But most of the antibody testings are used for epidemiological purposes in order to measure herd immunity. And for donation of convalescent plasma, I think Dr. Jayasinghe is going to talk more about convalescent plasma. And for Kawasaki-like syndrome, we can confirm the diagnosis of past COVID-19 with antibodies. So those are the three areas that we use the neutralizing antibody test. Now, new CDC guidelines are out for retesting. Should we or should we not retest a patient within 3 months? Now, the studies have proven, NIH studies have proven that retesting a patient within 3 months is going to be useless. So please don't order these tests within 3 months, because there's a high chance that it can be positive again and again and again. That does not mean a persistent infection. That only means that there are viral particles, viral RNAs that are emanated that you're going to catch with your nasopharyngeal swab. So that is very clear cut now. And then discontinuation of quarantine after 10 days for most of us. And in the hospital also, for most of the patients who are mild to moderate symptoms, with no fevers, no symptoms, we're going to discontinue isolation after 10 days of the present onset of clinical symptoms. Exceptions are the ICU patients, those who are on the ventilators, we're going to go to 20 days. Why? Because they could continue to shed the virus, especially the immunocompromised patient with poorly controlled diabetes, HIV patient, your transplant patients who are on immunosuppressives. So these guidelines are very clear cut. Management of COVID-19 are very, very, very streamlined now. Supportive care for most of us, as Prof. Walter Jayasinghe mentioned. But for majority of patients, we send them home with high-dose vitamin C, vitamin D3, and zinc sulfate. Dexamethasone have been brought into the picture because of this recovery trial. But even before the recovery trial by Oxford University, we use dexamethasone only for the middle and the leftmost cases that we said. That's the pulmonary stage, and then people who are in overdrive immunity or cytokine storm. So patients who are hypoxic, we are going to give low dose dexamethasone, 6 milligrams once a day, intravenous or oral, for 10 days. And that has shown mortality benefits. The death rates have gone down by 35% in ventilated patients and 20%. So, and remdesivir, we give remdesivir to a lot of patients nowadays. Those who present to the hospitals, they present to the hospitals for a reason. We admit the patient from ERs for a reason. So those are sick enough to receive remdesivir. Unfortunately, remdesivir is an antiviral that should be given in the first 7 to 10 days, because that's a viral replication stage. But a lot of patients present to us afterwards, they're not going to benefit much from remdesivir. So the NIH study has shown that remdesivir had cut the replication that way, enhance the recovery rate by 30%. And hydroxychloroquine, Prof. Walter Jayasinghe talked about hydroxychloroquine. We still use hydroxychloroquine, but this medication has been highly politicized in this country. Therefore, we stay away from it. But your mild to moderate patients, non-hospitalized patients, mild to moderate symptoms, hydroxychloroquine can potentially be helpful. It is an anti-inflammatory agent, but it is used as an antiviral agent, as studies have shown. There have been a variety of studies that have shown good and bad effects of hydroxychloroquine. Tocilizumab is for hyperimmune stage, hyperinflammatory stage, those who are in cytokine storm. Unfortunately, that's kind of too late in the game, but we still save their lives, but their mortality is still high. But this is a medication that we have been using, IL-6 receptor blocker, which is a monoclonal antibody that we use. But a recent study from Roche, the results from a recent study was not very encouraging to us. Very expensive medication. Other COVID therapeutic trials have been going on. And the latest one from Eli Lilly company called baricitinib, BARICITINAB. So baricitinib is again an anti-cancer, anti-inflammatory medication that inhibits JAK1 and 2 genes. That has been tested in a study that we call ACTT2 trial by Eli Lilly in combination with remdesivir. So we have shown that remdesivir baricitinib combination cuts down enhances the recovery time. HIV medications such as Kaletra have been used, especially in India and other parts of the world, with some success. Influenza medications such as Tamiflu, oseltamivir, favipiravir, and then baloxavir, which is which are being used for influenza in this country and some other countries. The most exciting aspect of new armamentarium is antibody cocktails or antibody medications. And there are two companies, Regeneron and Eli Lilly, who have been considered for FDA approval after they are in stage, I believe, phase 3 trials right now. There are a whole bunch of other medications such as colchicine, the gout medication, remdesivir inhalational form, beta interferon inhalational form, and most excitingly, Pepsid, your famotidine H2 blockers have been used in this situation with some promising results. And I use, and most of the hospitals use ivermectin, which is a very inexpensive anti-parasitic agent to treat mild to moderate hospitalized patients because it's very inexpensive and studies have shown some in vitro promise. Thank you so much. I'm gonna stop sharing my slides and then move on to our next speaker, Dushyant Jayavira, who's a professor of medicine from an infectious disease specialist from the prestigious University of Miami. And over to you, Dushyant, to tell us more about
Thank you. Thank you, Deepti. It's a wonderful presentation, Deepti. Thank you. And I want to thank the Sri Lankan Foundation for inviting me. Just to make a comment, I'm honored to be with Anil Jayasinghe and Saroj from Sri Lanka, and I'm so honored to be part of this panel along with them, because it's amazing what Sri Lanka did during the pandemic. The two things, in addition to what Dr. Jayasinghe mentioned, I think, in my opinion, it's the free education that we provide with high literacy rate, and also the free healthcare makes Sri Lanka a unique combination of excellent healthcare system with a highly high health literacy rate, which leads to good outcomes. Now, just to put things in context, if we compare Sri Lanka with the 21 million people with state of Florida, the state of Florida, the number of cases was 683,000 as of today, compared to 3,300 in Sri Lanka. In state of Florida, there were 13,300 deaths compared to less than 50 in Sri Lanka. So you can see that we have to be very proud about what our achievements were in Sri Lanka. Now let me get on to my slides. Okay, so this slide shows on the left-hand side the Sri Lankan Medical School where I attended, and I was there in February when Saroj invited me to give a couple of talks, and it's an amazing institution which I have great gratitude. On the right is at the University of Miami, where I work. When I step outside, this is what I see. So my talk today is going to be on convalescent plasma and vaccines. Next slide, please. Next slide. So let's talk about the background and the justification for convalescent plasma. So convalescent plasma, the definition is that basically somebody gets infected, and then when they recover, you take the plasma because that has all the antibodies that you have developed against the virus. So now you can use those antibodies as passive immunization of a newly infected person so that you can neutralize the viruses. Next slide, please. So what's the history? What's the history of the use? Since 1892, until the antibiotic era, which was like 1928 when penicillin was discovered by Alexander Fleming, the serum therapy was used to prevent and treat many bacterial infections like diphtheria, pneumococcus, measles, mumps, you name it, they used. The specific immunoglobulins also have been effective. I think Deepti mentioned that. And later, when the Argentinian hemorrhagic fever happened in last century, in the 70s, in Argentina, with 90% death rate, this was used. But the funny thing is that there were no randomized proper controlled studies about to look at the efficacy or not. Next slide, please. So continuing the same, even during the bird flu epidemic, this was used. And the question is, why do we use convalescent plasma? The reason is that in a pandemic situation, in the absence of a vaccine or an antiviral drug, or all the other things that you need to control it, with high mortality, this is something that any little hospital could make and use it. So convalescent plasma was a very convenient, rapid use therapeutic modality, which you could use in a pandemic or epidemic situation. Next slide, please. So what was is it safe? The question is it safe? Yes, of course. The Mayo Clinic, funded by the NIH, did transfuse about 100,000 patients. And in Florida itself, we have given over 10,000. And the transfusion reactions are less than 1. The thromboembolic events are less than 1. And the cardiac events are high, but that's because COVID causes endothelial dysfunction in vessels, which leads to cardiac dysfunction. So the question you beg is the answer is, why did we transfuse 100,000 without doing a randomized clinical trial up front? I think the justification is that there was a mandate from the government to quickly do something to save lives. And it was a kind of Hail Mary pass or a throw in the kitchen sink to save lives. Next slide, please. So on August 23rd, the FDA commissioner wrote to the government saying, I have concluded that emergency COVID-19 convalescent plasma use is justifiable. And he covers his backside by saying convalescent plasma may be effective in treating COVID-19 because there's no randomized controlled data. It says that there's no adequate or approved available alternative for emergency use of COVID-19. So basically saying that we don't have anything. Maybe remdesivir works, but not in every case. And if remdesivir has to be given in IV use, and there were some toxicities involved in that. Hydroxychloroquine works, but sometimes you have toxicity. This is fairly safe, but there's no data. So he goes on to say his well-controlled randomized clinical trials are needed, although I have said that it is approved for emergency use. So basically, we are caught in an awkward situation where we have to run a controlled clinical trial, but we are also obliged to give patients this drug. Next slide, please. This therapeutic modality. So when we looked at the data, as physicians, we go by evidence-based medicine. So we want, when we say something, that it is based on evidence. And you have to be very, very clear about it. As physicians, 50% of the stuff we say, after a couple of years, you realize that they are wrong. And we don't know what that 50% is. So one thing I teach my medical students about humility training is very important to be humble, because we don't know what we say is right and what we say is wrong. So how you hedge the bets is to get as rigorous as possible clinical trials, and then you analyze and say, okay, based on the clinical trials, the data looks good. So we looked at Cochrane database and looked at 20 studies. Next slide, please. So basically it looked at all the hyperimmune serum and use of convalescent plasma and asked the question, is it really effective based on published data? So first point it comes off is that the overall risk of bias is included in studies. So there's fundamentally bias because some studies are not randomized, some studies are not blinded. So if you have to be absolutely certain, you're looking for studies that are blinded, randomized. Outcomes are very well-defined outcomes, and they are statistically rigorous and with no conflicts of interest. So final outcome of this review is that we are very uncertain whether convalescent plasma has any effect on all-cause mortality. It means any cause of death at hospital discharge. And it says very uncertain whether convalescent plasma prolongs time to death. And he also says we are very uncertain whether convalescent plasma has any effect on improved clinical symptoms at 7 days. Then you ask, what on earth are we doing with this convalescent plasma if this is what the data shows? So now we are still struggling because the FDA says, give it, and we are asking how and why. So whenever you have a conflict like this, you go to the ethicists. Ethicists are people that, if you put 20 people in a room, you get, 5,000 opinions. So, but we went, next slide, please. We looked at the, sorry, before that, I want to talk about this slide. So meanwhile, the thought leaders were questioning, is this really right? For example, Rob Califf and Mark McCallum, all these guys are FDA commissioners. And Walid Gellard from Center for Pharma Research from Pittsburgh, and then people from Scripps and WHO says, guys, we have to be careful with this. So next slide, please. So we went to one of the review articles written by the American Journal of Bioethics, asking, how do you ethically give this with a straight face and understanding we are doing the right thing? So at the end of the day, no matter how big the research is, whatever you do should help the patient. I mean, that's your moral obligation. So the first principle of EUA allocation to be minimize harm. So I shouldn't give convalescent plasma if it is causing any problems. Now we know that I showed the data out of almost 100,000 infusions. The side effects are very minimal. So it's pretty safe. It's pretty safe. I mean, right now, I'll talk about it a little later. So it should be given to people who are sick when no other options are available. I mean, we are questioning that because, ethicists look a different view, but we are looking at from the biomedical standpoint, we think that, well, we should be giving a little earlier. If you assume without evidence that convalescent plasma works, and giving it freely does not minimize harm when there is scientific equipoise. So that means that if you're not sure whether it works, just giving everybody willy-nilly is not going to help people. It's not generalizable knowledge or you're not going to improve the outcome. So you have to be very careful. Just because there is no treatment, just giving ineffective treatment is not going to make anything good. So fundamentally, the ethicist also says it should be given only where places there are no randomized controlled clinical trials. But if there is RCT, then you should give RCT. So that's how we justify one of the studies we are doing. Next slide, please. So these are studies funded by NIH, which is, I am the PI for University of Miami. We run it with NYU, Einstein, and Yale. And basically taking the people who are hospitalized within first 3 days or 7 days from the time of symptoms and giving them a randomized controlled trial, completely placebo-controlled, nobody knows. And looking at the WHO ordinal scale, 11-point scale, and we are looking at the improvement of 11-point scale. So this study is ongoing. Unfortunately, our or fortunately for the people, the COVID epidemic is slowly dying down, but still we are continuing to enroll, and we just transfused one patient yesterday. So now you can see that the importance of studying things properly, doing things properly, leads to good information. Next slide, please. So what does this convalescent plasma really mean? So in this slide, you show that on the top right side, the curve graph shows that when you go up to about 60 days, you get a maximum amount of antibodies. Again, the top right-hand graphs here, more than 42 days, you get the highest antibodies. So when you give convalescent plasma, in my study, what we do is that we take plasma, which has in the small table at the bottom, the IgG and the receptor binding domain specific IgG. We look for high concentration of receptor binding domain specific IgG and specific IgG high levels. And those are the ones that we give. So that way we are comparing apples to apples, where everybody gets high receptor binding domain specific IgG and non-specific IgG levels, and they are compared with placebo. And now at the end of the study, we look at the results and we can say, if you give people with high antibody titers, then you get a good outcome. So that's the question we are trying to answer. Next slide, please. So in summary about the convalescent plasma, historically it has been used. The FDA says go and use it, but only in the life-threatening conditions. And we are thinking, wait, we need some more data. Cochrane says the data is really bad. You need to do decent studies. So now the randomized controlled trials, they're run by NYU and us leading one, and then there's one run by Vanderbilt. So we are running, and Hopkins are running 2 other studies. So we will get an answer within the next couple of months. Next slide, please. Now let's talk about the COVID-19 vaccine. Now, before I get to that, I looked at asking, what happens if we say tomorrow we develop a COVID-19 vaccine? How are you going to operationalize this? Who's going to get it? How are you going to manage this? And who will accept it? In the United States, there's a fair amount of lobby who doesn't want to take vaccine. I mean, it's amazing that even I was doing a clinic yesterday, or not yesterday, on Friday, and 3 people refused flu vaccine. And I was like, I don't have any hair, but if I had hair, I would have pulled them off, because saying, what is this? What is this? So next slide, please. So I went to UNICEF data warehouse this morning, actually. I pulled down the data and filtered it and tried to compare Sri Lanka, Costa Rica, India, and USA, and looked at the basic vaccines that we give. So Costa Rica is considered like the equivalent of Sri Lanka from the WHO standpoint, high literacy rate, good healthcare outcomes, decent free healthcare, and then comparable. And we are leading people in the more middle income countries in the world. And it's interesting. We seem to be leading among everybody else. Definitely better than India, better than Costa Rica, and even better than USA. So I think we should be, I think it comes back to what Dr. Anil Jayasinghe and Dr. Saroj Jayasinghe was talking about, the commitment of the government, the response of the people, and the amazing healthcare system that comes together in a small country. And we are a model of healthcare to the rest of the world. The only thing that worries me a little bit is that how did we get 99 on all of those? So that's why Saroj and Anil are here. I mean, perhaps they could go back and say, look at a little bit of data integrity and see whether there's somebody, crossing the T's and dotting the I's and making sure that this data is really clean. Because at the end of the day, data integrity is really important. I trust Sri Lanka, but it's worth checking and seeing whether these were all nicely collected. Next slide, please. So let me briefly, before I get to the vaccine, walk you through how the virus works. As you know, as Deepti mentioned, that the virus enters through the lungs, and there are these angiotensin 2 receptors which binds on to the spike protein, and they gets internalized, and then the virus replicates, and the virus comes out. And the virus gets picked up by the antigen presenting cells, and it leads now to either T cell response or a B cell response, and those are the antibodies that comes out. So what we are trying to do is now to generate a vaccine that will create this antibody response without getting the infection. Next slide, please. Now this slide tells you like the timeline. So if you start from the bottom, polio, it took 60 years. It took 60 years to develop a vaccine. For Ebola, it's around 15 years. For what's a COVID-1, or no vaccine, nobody bothered. MERS, no vaccine. And SARS, COVID-19, within 12 to 18 months. So when you ask why, first of all, how did we do this? And why is that this is done so fast? First of all, if a disease affects Europe and America, it gets solved very quickly. If HIV, I'm a HIV guy, if HIV happened being only in Africa and India, nobody would have taken notice of it and we would have got wiped out completely. It had to affect the Western world for the people to start spending money. So the first thing is you need money. The second is that you have to ask is that, now things like polio, they went through the regular channels. So people are kind of worried saying, oh, if you squeeze everything together, would you compromise safety? So sometimes you could take a contrarian view and say, maybe this is the way we should be doing things. You don't need all this other million animal testing the answer will lie later on as we follow these people. When you do phase 4 studies, phase 4 is that once you approve, you track these people and see what side effects happen and what happens. And then we will know. But maybe this is the way to do things. Maybe that we should re-engineer the entire drug development system and say, we are doing too many unnecessary things. Let's cut out nonsense and get to the heart of it. So next slide, please. So in the development cascade of vaccine development, I think the first cases of COVID-19 was around December 2019. The Chinese released the genetic sequence first week of January. So what we do is that we look at the genetic code and we look at definite targets that we want to block or generate antibodies. So you prepare in small batches and you test in vitro, see whether you can see some neutralization. If you can't use the virus, then you can use just spike protein and use that. Now these things have to be done in biosafety level 3 or 4 labs. Very few places in the United States have biosafety level 3 or 4 labs. Not even certain parts of the National Institute of Health. When this came up in Miami in January, I told the president that we should have a BSL-3 lab. He said, yeah, it's a great idea. So it's still a great idea. Some places have, for example, Case Western and Nebraska, and few places have UNC in Chapel Hill has a BSL-3. So these things need a special labs to do because these are highly infectious. So once you develop this target selection, then you have to do a target validation. You check the target, you test in vitro. Now you validate it with preclinical animal tests. And then the animals are working well. Now you move it to the phase 1. The phase 1 is that you give to healthy humans, healthy bodies, very few people, and see for any side effects. If the side effects are okay, that's just the safety. So safety is absolutely paramount. And then you go on to efficacy. You look at the antibody response. You're not only the safety, now you're looking at the neutralizing antibody increases. And then you go to phase 3, where you're looking at efficacy, that's antibody response, and also efficacy with clinically, whether there's a difference between getting the vaccine versus not getting the vaccine. So if there's no lack of efficacy, the drug is gone. If there are too much side effects, the drug is gone. If both are okay, now you become stuck in worldwide distribution. These things have to be sent at minus, it's minus 100 Fahrenheit degrees. So you have to be really cold so that you don't, so you have to have systems to distribute this across the world. You have to have manufacturing capabilities and the supply chain and all that. So that is the complicated life of vaccine development. But it can be done. It doesn't have to take 15 years. I think 18 months, 2 years, looks good. Next slide, please. So now the COVID-19 vaccines, you can see there are 25 in line. We have 140 a couple of months ago. Now it's down to 25. There are 2 approved vaccines. One is the CanSino Biologics from adenovirus AD5 from the Chinese military. And they're going to run some phase 3 studies at Saudi Arabia and Pakistan. It's been approved to use in Chinese military. And at least on the phase 2 studies on animals, it had an excellent immune response. In fact, one of the reviewers who compared this with Oxford said the Chinese vaccine had even a better response than anybody else. The second one is Gamaleya from Russians. I mean, they don't tell us what they do, but they had the idea. They put 85 and 826 two adenoviruses as a vector. And the problem is that if you combine too many adenoviruses, you may get a adverse response because vaccine-induced adverse response, which sometimes you can have an opposite effect of the vaccine. The vaccine is done to decrease the side effect. If you get the symptoms or complication of coronavirus, but you can have the opposite due to Th2 response of immune system. So we're not quite sure how this. They don't tell us too much about Russian vaccine, and they're going to send some things in Brazil and Mexico. Next slide, please. So currently there is this AstraZeneca Oxford vaccine, then you have the Pfizer, then you have the Moderna, and I'm leading the Janssen vaccine study. We're hoping to vaccinate the first person in next Monday, hopefully, God willing, if things okay, not this Monday, the following Monday at University of Miami. Next slide, please. So, next slide, please. So I'm going to introduce something called, next slide, please. Okay, I'm going to talk about this thing called challenge vaccines. So the history of challenge trials. So first of all, let me give you a brief history about Edward Jenner. So Edward Jenner in 1796, he deliberately exposed the gardener's son to cowpox, and then exposed the son after some time to smallpox, and the kid didn't get it. Now this will never ever go through our IRB, but they didn't have IRBs at that time in 1796, right? And also in 1796, vacuolation, that means that, this scratching with the infection was a tradition that happened at that time. So Jenner, this is not really Jenner's original idea. There are a lot of people who are doing that. In fact, they say that, hundreds, during the ancient, the Turks used to do that. So this has been done by many people, but Jenner did it scientifically, as far, if you can't call it scientifically. And he didn't, the kid didn't get it. So almost two centuries later, we managed to control smallpox. So this is the concept. You vaccinate and you deliberately expose. So the first influenza challenge study was done in 1937. In 1980, the University of Virginia did a challenge study, giving the vaccine and exposing them to deliberately to influenza A and B. And that's how they developed Tamiflu. You can see that 2009 H1N1 pandemic, NIH did the same thing with 46 people. In 1976, NIH did this challenge study, they found that the vaccine didn't work by doing the study and saved a lot of lives. Next slide, please. So as you can see, malaria, typhoid, dengue, we have done this. The question was, why not for COVID-19? Why not vaccinate selected people who know exactly what the side effects are and then get exposed? And the Imperial College of London said that if you do that, you could save millions of lives by doing, mathematical modeling. They said that you could save about, by prediction, millions of infections. And there are 30,000 people in the United States who have agreed to get vaccine and then get exposed. So then you look at and ask, so who would you expose? The best people to expose is between the ages of 18 to 30. They are the best group who are healthiest and has the lowest mortality and least risk. So obviously they shouldn't have any comorbidities like obesity, diabetes, hypertension, immune deficiency, all that. If you take all that away, 18 to 30, if you do that, you could get an answer very quickly. I wonder whether Chinese and Russians did that. Next slide, please. So as I said, I'm not going to talk too much about, because Malini is going to talk about it. I'm going to skip this. The only thing is that safety is the primary goal. And you have to use the correct adjuvants or things that boost the effect to drive the response to what you call the Th1 response, not Th2 response, because if you get a Th2 response, then the side effects bump up. Next slide, please. So in conclusion, many vaccine platforms are pursued and likely to be delivered next year. And I'm sure there are more than one winners. And universal vaccine against other zoonotic coronavirus is likely to be challenging, and you have to come up with different vaccines. Next slide, please. With this, I'm going to end. These are my disclosures. I want to thank Deepthi and Walter Jayasinghe, doctors Deepthi Jayasinghe and Walter Jayasinghe, and the Sri Lanka Foundation, and thank you. And I'll hand over to Malini, who is the next speaker.
Thank you for speak today. Can you hear me?
Yes.
Okay, wonderful. So like Dr. Jayawira said, I'll be speaking more about vaccine safety and not specific concerns about the safety signals such as the transverse myelitis that's come up with the AstraZeneca trial that's been widely published, but more about general vaccine safety and what's being done, particularly post-approval for vaccines. So it's widely accepted that access to a safe and effective COVID-19 vaccine is a critical component of the public health response to the pandemic. And Dr. Jayawira discussed development timelines for typical vaccines and the COVID-19 vaccines. While the COVID-19 vaccine is on track to become the fastest developed vaccine in history, this expedited development timeline has many people worried about whether critical steps are being skipped. So today I'll discuss some of those safety concerns that have arisen, as well as discuss some of the post-licensure safety monitoring that's routinely performed for any vaccine. So since the SARS-CoV-2 genome sequence was first published, the race to develop the vaccine has been underway. Different vaccines have started human trials throughout the world, and the use is expanding beyond clinical trials, as was discussed in the last presentation. In Russia, China, UAE, they've all had vaccines that are now outside of the clinical trial sphere. There's widespread concerns that the vaccine approval outside the scope of clinical trials may be premature. In the US, the goal to have a safe and effective vaccine available by January 2021 or sooner has been bolstered by funding from the Department of Health and Human Services as part of the Operation Warp Speed. While the FDA has issued guidance providing clear recommendations about efficacy thresholds, populations for trial inclusion, and safety considerations prior to the initiation of most large-scale COVID-19 vaccine trials, many remain skeptical that the rush to make a vaccine available threatens the ability to appropriately study the vaccine and vaccine safety. Some of the implications of rushing a vaccine include lack of safety and efficacy data in populations that are typically excluded from clinical trials, such as those over the age of 65 years and pregnant females. Multiple studies have shown that these groups are at higher risk for severe COVID-19 disease and associated complications compared to other groups. Pregnancy is a particular concern for clinical trials because at this point in time, there haven't been any pregnant adults included in these trials. The potential risks for COVID-19 vaccination in pregnancy are largely unknown because of this. So once licensed, healthcare workers and others frontline essential workers who are likely to be the first targeted for vaccine will be including women of reproductive age, and inadvertent exposures during pregnancy may occur. So it's with these inadvertent exposures that we'll be studying much of the pregnancy complications or side effects. So it's not as part of the clinical trials thus far. There are also questions about the chosen study endpoints, as data on immune response alone is not a generally accepted basis for approving a vaccine, because immune response may not be directly proportional to the degree of protection against the targeted infection. While the FDA has set recommended efficacy thresholds, these recommendations are not requirements, and deviations from these may occur. Additionally, if a vaccine is made available before appropriate results about vaccine effectiveness are available, People who receive the vaccine may have an unreasonable expectation about their risk and stop using other preventive strategies, such as wearing face masks and keeping appropriate social distance. Premature cessation of these other prevention strategies may result in worsening of the pandemic. Finally, if a safety concern arises in the follow-up period for vaccine trials, but after initial fast approval has been granted, this may impact the public's trust in vaccine development process in general, with extension to concerns about safety of other routinely used immunizations. As Dr. Jayawira noted, there are currently 9 vaccines in phase 3 clinical trials. The minimum recommended number of participants for phase 3 vaccine trials is at least 3,000 participants, and most of the COVID-19 vaccine trials have a goal of recruiting around 30,000 participants. All the current phase 3 trials are placebo-controlled, meaning that the participants will be randomized to either receive the vaccine or to receive an injection containing a placebo. Participants will then be followed for a varying length of time to evaluate for both adverse side effects and COVID-19 disease. Adverse side effects will include both acute side effects such as fever and injection site reactions, and longer-term side effects which may take weeks or months to develop. The research teams will then compare rates of adverse effects amongst the COVID-19 disease between the participants who received the vaccine and compare those to those that received the placebo. In the United States, the FDA has set the current vaccine effectiveness threshold at 50%. This means that an individual who is vaccinated would be 50% less likely to get COVID disease than individuals that weren't vaccinated. Federal officials have recommended that at least 150 COVID-19 infections must be recorded among trial participants, with at least twice as many occurring among the placebo group. However, earlier this week, Moderna reported they'll seek an emergency use authorization, or EUA, if early results from their clinical trial show a 70% effectiveness after 53 COVID-19 cases have been documented among trial participants. Their spokesperson said that they would focus their EUA on high-risk groups such as healthcare workers who are at higher risk for disease exposure and the elderly who are at higher risk for severe disease. It's important to remember that because vaccines are given to healthy people by choice, which is unlike medications and treatments that we've heard about earlier, like remdesivir and convalescent plasma, that are given to patients with known disease, vaccines reach a larger number of individuals than other therapeutics. Because of that, it's possible that certain rare outcomes may not come to light during clinical trials. Even if an adverse effect occurs in a limited number of participants during the clinical trial, once a larger number of individuals is vaccinated, we may see higher occurrence of these events. For example, if one patient has a severe adverse event during a clinical trial and it's found to be related to the vaccine, if the study population is representative of the population, the larger population that will eventually be vaccinated, once it's been widely distributed, we may see thousands of similar adverse events. It's also important to remember that rare events may not occur during a clinical trial, and only after a large number of individuals receive the vaccines do these rare events come to light. Because of the ongoing concerns of adverse events following immunizations in the United States, there's programs which provide monitoring for vaccine safety after a vaccine has been licensed. 3 of these programs in the US are the VAERS program, PRISM, and the VSD. I'll just give a brief description of each of these in the next couple of slides. So the Vaccine Adverse Events Reporting System, or VAERS, is a national vaccine safety program run by CDC and the Food and Drug Administration. VAERS serves as an early warning system to detect possible safety issues with US licensed vaccines by collecting information about adverse events, possible side effects, or health problems that occur after a vaccination. VAERS was created in 1990, and anyone can submit a VAERS report. These reports are then compiled to detect possible safety problems called signals that may be related to vaccination. If a vaccine safety signal is identified through VAERS, then scientists will conduct further studies to find out if the signal represents an actual risk. And one of the programs that does these additional studies is the VSD, which I'll talk about in a minute. The Post-Licensure Rapid Immunization Safety Monitoring System, or PRISM, is part of the Sentinel system, the FDA's national system for monitoring licensed medical products. PRISM is specifically focused on vaccine safety and uses health insurance claims database to identify and evaluate possible safety issues for licensed vaccines. So this isn't reliant on patient reports or provider reports. This is strictly limited to claims data, and the studies that are done through this PRISM program are designed to be able to look at specific timeframes after a vaccine is given, and then the follow-up ICD-9 or 10 codes that are used to identify the specific adverse events they're looking for. The Vaccine Safety Data Link, or the VSD, is a collaborative project between CDC's Immunization Safety Office and 9 health care organizations around the United States. Health Partners, which is the organization for which I work, is one of those healthcare organizations. The VSD uses electronic health data, including both medical records and insurance claims from participating healthcare organizations to conduct vaccine safety studies based on concerns raised from the medical literature, rapid cycle analysis of its own data, and reports to VAERS. In this way, the VSD complements the VAERS reporting system as a safety signal identified by VAERS can be more thoroughly investigated using patient-level data. So that's an additional thing that the VSD offers in addition to PRISM or VAERS. We have that patient-level data that's available so we can do further investigation, including things like laboratory results and testing and actual chart reviews to try and validate outcomes as well. Specifically, the VSD attempts to monitor possible adverse events when new vaccines are licensed or when there are new vaccine recommendations. So in conclusion for this, and I'm happy to answer questions later, but I thought it would just be a briefer presentation. It's possible that the COVID-19 vaccine will be available before phase 3 trials have completed their long-term follow-up. The FDA has issued guidance for COVID-19 vaccine research standards, and vaccine safety monitoring will continue through both clinical trials and by other programs after a vaccine has been approved. So it's not to necessarily completely allay concerns about the fast pace of this, but the vaccine safety monitoring will be continuing for a long time to come.
Thank you so much, Malini. And that was an excellent presentation. And over to our last speaker, Dr. Davinder Kurera, who's in California, Southern California with us. He's an emergency room physician in San Dimas area. He's gonna talk more about emergency room guidelines and protocols for COVID-19. Go ahead, Dave.
All right, thank you very much. First of all, I'd like to thank everyone for giving me the opportunity to give the emergency room perspective on COVID-19. I do want to acknowledge that really it's the doctors and especially the nurses on the inpatient side of the house that really do a lot of the hard work with these COVID patients. They're the ones dealing with the really sick patients and really the long-term exposures to these COVID patients. So we're very grateful for all the work they do. From the ER perspective, you know, our biggest role is the triage process, deciding who will need to be admitted to the hospital or who we can send home to ideally isolate and hopefully get better on their own. So to that, I looked into what are our guidelines for admitting patients. So I looked at my governing board, the American College of Emergency Physicians, or ACEP, and to see what their recommendations were. And they have worked with a company, Evidence Care, to create a triage process for emergency physicians. So this will classifies patients from a mild to low risk to critical. And it uses 7 categories of measurements. These include a QCSI calculator, which is a predictive model of early hospital respiratory decompensation among those patients with COVID-19. This PDF can actually be found at the ACEP website, or there's an interactive version of the pathway at the Evidence Care website. But personally, I find this a little too cumbersome. I noticed, you know, they used clinical gestalt, or essentially the doctor's opinion, as one of the criteria on who can be discharged home. So I looked further into what ACEP recommended. And they also talk about this New York Presbyterian COVID-19 evaluation pathway. So this was developed across the campuses of the New York Presbyterian Hospital system. So these were the systems when New York was hit hardest with COVID that were experiencing a lot of these COVID patients. And one advantage of this pathway is that it was able to incorporate the real-time feedback from those different institutions. So it divides the patients into 3 categories. The low risk, they have clinically mild illness, typically normal vital signs. These patients are usually under 50. And most importantly, their oxygen saturations are greater than 94% and their respiratory rate is less than 22. So what we'd expect in a normal patient. They recommend from that point doing an exertional O2 saturation. So you ask the patient to walk a minute in place at bedside and see where their oxygen saturation goes to. So if it's greater than 90%, they recommend essentially no routine chest x-ray unless you're concerned about another pneumonia or something like that. And no viral testing needs to be done on these patients. Now, if the O2 saturation ends up dropping below 90% in these patients, they recommend going to the moderate risk evaluation. So these patients, their oxygen saturation usually is between 90 and 94%. They have a higher respiratory rate greater than 22. And they'll be, a lot of times you'll factor in their age over 50 or with comorbidities like hypertension, COPD, diabetes, obesity, cardiovascular disease, lung disease, cancer, the immunocompromised, and those with renal disease. They do recommend obtaining labs and imaging with these patients. So your basic your CBC and your comprehensive metabolic panel. They do recommend doing the SARS COVID-2 PCR testing on these patients. And then other tests include CRP, D-dimer, ferritin, lactate, LDH, and troponin. And those are really used by, if they end up being admitted, the inpatient team to further risk stratify those patients. If there is no indication for admission after a period of observation with these patients, they may be discharged home. And they talk about with these patients, in order to ensure good follow-up, that they have a working telephone. So if the test results come back, you're assured that you can contact the patients, and also that they have assistance at home to limit their exertion to prevent that oxygen desaturation, and to ensure that they have good follow-up, either with a telemedicine or follow-up at a clinic. You should consider with these patients sending them home with a pulse oximeter so they can keep track of that oxygen saturation, or supplemental oxygen. So if their exertional oxygen level end up being between 90 and 92%, you should strongly consider discharging them home with supplemental oxygen. Now, if that's not available, we really should consider admitting those patients to monitor them closely in the hospital. And then finally, the high-risk category. These are your critically ill patients. They're oftentimes in respiratory distress, so they'll have significant tachypnea or their work of breathing or hypoxia on the O2 monitor. These patients may often be in shock, so their blood pressure is low, their heart rate is fast, or you see evidence of clinical deterioration while you're observing them in the emergency department. Those patients definitely need those labs and imaging that we talked about before, a consult to critical care, and then the use of supplemental oxygen to keep their saturations up. So non-rebreather or nasal cannula or both oftentimes will help. And then we talk about if a negative pressure room is available, starting high flow nasal cannula or BiPAP. And I'll talk a little bit more about those in a couple of slides. One big part of emergency medicine is helping taking care of that airway, especially in an emergency situation. So considerations with these patients, the asthma patients who we consider COVID in, talk about using meter dose inhalers versus nebulized medicine treatment. We found that to try and avoid nebulized medical medicines, like nebulized albuterol treatment for asthma, mostly because of the fear of contamination to the staff and to other patients. So whenever possible, using a metered dose inhaler is preferred. And then with the airway, we have BiPAP and CPAP, BiPAP being bilevel positive airway pressure, and CPAP continuous positive airway pressure. And we use these oftentimes as a bridge to help avoid intubation. Especially in the COVID patients, we found that, that those that get intubated tend to not do as well. So we want to try and avoid getting to that point, seeing if we can get them over that hypoxic period without doing the intubation. Use is controversial in patients with COVID, mostly because there's a theoretical risk of increasing aerolization of the virus and increased exposure and risk to the healthcare workers and other patients in the department. The Society of Critical Care Medicine does support its use, but they give the caveats that patients must be monitored carefully and intubation should not be delayed if there's any signs of worsening. And ideally, it should be in a negative pressure room. So that's where particles aren't leaving that room and potentially infecting other patients in the department or other staff. High flow nasal oxygen. This has been associated with lower mortality in hypoxic respiratory failure. So especially when compared to conventional oxygen therapy, there has been a decreased risk of subsequent intubation and a need for ICU admission in these patients. The degree of aerosolization has been shown to be minimal with high flow nasal oxygen. So it's now become the recommended as the oxygen therapy of choice in patients with respiratory disease. You may also consider having a surgical mask placed over the patient when treating them with high flow nasal oxygenation to help mitigate the exposure to others. And the medical staff should always use airborne protection, including N95s or PAPRs, and treating in the negative airway pressure room. As far as imaging in these patients, CDC doesn't currently recommend chest CT or chest X-ray as a diagnostic method for COVID-19. They say viral testing remains the only specific method of diagnosing, and a confirmation test is required even if radiological findings are suggestive of COVID. I've actually personally had a patient who came in with abdominal pain. I didn't have suspicion of COVID in her. Got an abdominal CT to work up that pain. And they, on the lung windows, they found evidence of ground glass opacities, which they considered suspicious for COVID. Subsequent testing of this patient did demonstrate that she was COVID positive. So, you know, there's always those incidental findings that imaging can help with. Chest x-rays in these patients, the findings are very nonspecific and they overlap with other infections, including influenza, H1N1, SARS, and MERS. Typically, there's unilateral or more likely bilateral infiltrates on their chest x-ray. They'll find consolidations. It'll oftentimes be multifocal. And pleural effusions tend to be rare in these patients. The CT findings, ground glass opacities or consolidations are the primary findings on CTs. It's bilateral is the most commonly seen. Though interestingly, the ground glass opacities are more likely an intermediate to late finding in the disease process. That's all I have, and I'll open up for questions.
So we are open for question and answer. We don't have much time anyway. The first question is from Aloma Ratnasoma, who is asking about the serious side effect that transverse myelitis is the name of the side effect that a patient from England experienced. Anybody wants to comment on that?
I can do that. So the vaccine has the transverse myelitis. There is a tendency for a neurotropism among the adenoviruses. In July, in July, they had another side effect like that with neurological symptoms with Oxford vaccine where they had to stop. And that time they investigated and it was multiple sclerosis. So it was unrelated to COVID-19, the Oxford vaccine. So they are investigating it. It is possible they are related, but you have to investigate further to be absolutely sure, because maybe that we will find that there's other reason for this transverse myelitis. That's what so far we know.
Sure. And there were a few questions about hydroxychloroquine once again. So I would direct that to Prof. Saroj at this point. If you're still here, go ahead and give us your experience with hydroxychloroquine, because it's a very, very politicized medication here.
Yeah.
Tremendous pressure to use or not to use hydroxychloroquine. Go ahead, Saroj.
Well, yes, thank you very much, Deepthi. They are using it in the hospitals designated for COVID. And as I mentioned before, it's being given to patients who have symptoms. At the initial stage, they gave it even to asymptomatic persons, but now they have limited it only to people who have symptoms. We were to do a trial on hydroxychloroquine using the cluster in the Navy base. Unfortunately, that we got ethics approval, we got everything, and we did the trial. But when we analyzed the cases, we found that a lot of them had not really taken the drug due to the adverse publicity. So, we just couldn't make any sort of statistical analysis. Yeah, thank you.
Yeah, that's unfortunate. So when you talk about randomized clinical trials conducted worldwide, there are a lot of unknowns about hydroxychloroquine. The biggest trial came from Henry Ford Medical Center in Michigan. They clearly showed that there were benefits, even mortality benefits, 35%. Like, that's a high amount. Mortality benefits from moderate to severe patients. But at the same time, Oxford recovery trial showed data on dexamethasone. So the thinking is that they use both medications, dexamethasone and hydroxychloroquine together. So we are not sure whether those numbers were absolute or not, number one. Number two, there was another big garbage study that came from, sorry for my French, garbage study came from Harvard University, where they borrowed some false data from a data vendor in Chicago that was proven wrong. And that actually, they convinced those professors, especially Dr. Mehta, convinced the WHO to stop using hydroxychloroquine and stop running clinical trials. So this is, in my mind, this is so unfair, right? This is a very inexpensive medication meant for poor patients who would have benefited from this, right? Specific categories are not mild patients. Mild patients do not need hydroxychloroquine or any other medication. All they need to do is to hunker down at home, take your vitamin C, the immune boosters, vitamin C, vitamin D3, zinc sulfate, and then hunker down at home, be in quarantine for 10 days or 7 to 10 days. If you can, sleep on your belly, because data has shown that self-proning, meaning proning or sleeping on your belly or on the sides, can potentially oximize the oxygenation. So mild patients, you don't need anything, but the moderate patients, right, or the patients who present to the hospitals, those are the people that we would have used hydroxychloroquine, and we were using it for 3 months with no problems before. But unfortunately, because of this tremendous pressure that they're applying, we kind of backed off, and FDA took out the EUA authorization. So now it's actually an off-label medication. So we still use off-label medications.
I have a comment. So we are running a study with Duke, funded by NIH, on hydroxychloroquine for healthcare workers for prophylaxis. Unfortunately, now nobody wants to take it. Initially we had targeted 15,000, but we lowered the sample size to 3,000. We are still short, and the problem now is that we can't enroll because people want to do the vaccine studies. So far the DSMB has looked at placebo versus the hydroxychloroquine for prophylaxis, and they are pretty happy with the safety. So it's unfortunate that all these politics muddle the whole the randomized controlled trials. Thank you.
That's right, Dushyant. Thank you so much. The next question is from our own Dr. Pujita Egudage, who's a primary care physician in Southern California. What are the recommendations for return to work in post-COVID patients versus hospitalized patients with test positive only versus close contacts? That's a good question. So the CDC guidelines are very clear-cut on this now. Return to work means discontinuation of isolation or quarantine, right? So mild patients, you can discontinue isolation in 10 days time. You know why it is 10 days? Because typically it takes about 4 days for you to present with the illness, right? After the exposure, it takes about 4 to 5 days for you to develop the disease. In other words, if you got the COVID-19 exposure about 4 to 5 days before, there's a higher chance of you being positive today if you are getting the disease, if you're contracting the disease. So leave 4 days for that, and then go ahead with for 10 days for the illness. So total of 14 days. This is a disease of 14 days. That's why they say after 10 days, you can discontinue the isolation, discontinue quarantine, normalcy, return back to work. Now, certain work situations require you to retake a test. We strongly discourage that based on the latest CDC guidelines from August 20th, last month, right? So the retesting protocol is very clear now. Retesting is very murky because the test results are all over the place. Once you're positive, there's a great chance that you're going to be positive for up to 3 to 4 months. That does not mean that you are infected. That does not mean that you are contagious. So if you don't have the symptoms, if you are past 10 days, you can get back to normalcy. Dushyant and Saroj?
Yeah, I think that's about, right. CDC keeps changing, but I think I agree with you. There's nothing else.
Malini, would you like to add something to that? A public health standpoint as far as return to work is concerned?
I think you've hit on all the points. If people are symptomatic, though, it really is until the fever has been resolved for at least 24 hours before you can even if they've been sick for more than 10 days. But I think the other thing for the asymptomatic patients that are just getting a test, and that's where it's been difficult, especially without knowing the CT threshold at which there is public health significance and transmissibility of the infection. So I think more work has to be done on that, and we have to determine whether or not they should be reporting out those the cycle threshold within the PCR testing, so that we have a better understanding of how that CT value corresponds to transmission within the community. So I think there's still more work that needs to be done with that.
Sure, thank you. So Malini, I think stay on with us. Is this vaccine safe for kids?
So children are another population. So under the age of 18, patients are not included in these clinical trials. And I think there has been a push to include them as well, because we have seen community spread linked to the pediatric population. And so it's hard to say if it's safe until we have inclusion of that population within the clinical trials, and whether or not that's going to happen, I'm not sure. I think there have been plans, and maybe Dr. Jayawira can say more about whether his study will include these different populations that aren't typically represented. But it is another area where we need to try and be more inclusive of the populations that should be receiving the vaccine. So we don't know at this point in time.
I think I agree with Malini. Unfortunately, our vaccine, Janssen vaccine study, we are starting people between 18 and 60, and then after that over 60. I think probably the phase 4 studies, they will include the children. The good thing about children is that the mortality and morbidity is very low, but the bad thing, as one of you suggested, is that they could get infected and infect the teacher. In fact, the last patient I put on a clinical trial with convalescent plasma was a school teacher who was 56 years old, got infected probably in school. I don't know whether it's school or not, but it's a school teacher. So, it's kind of, we feel really bad about the school teachers who are exposed to this possibility.
Yes, thank you so much. And then another question, this is a no-brainer. If you're anybody over 6 months of age, you should take the flu vaccine. So the question is, if I'm over 65, I've never taken the flu shot, never in my life, and I don't have any comorbidities. Should I still take the flu shot? Yes, definitely you should take it, because simply, right? Think about it, right? If you're doing a service, if you're doing a disservice to yourself and others by not taking the flu shot this year around, we don't want a twindemic. We don't want a influenza outbreak. Who knows what? Southern Hemisphere mitigated their influenza very well this time, thanks to the COVID mitigation steps. But America is different, guys. You know that, right? America is very, very polarized, politically and otherwise. So please stay. We don't know which strain will come here. We don't know whether there'll be another H1N1 scenario under the circumstances, right? So please take the flu shot, and time again, influenza vaccine, right? Time and time again, we have shown that this is a safe vaccine, and we have 4 different types of flu vaccines now. Of course, the common ones, intramuscular one, there's an intranasal one for non-immunocompromised patients, and then there's the high dose flu vaccine, and then the 4th for people with egg allergies. So please take your flu shot. Another question.
Before that, just to piggyback on that, especially from the ER perspective, this winter, we are anticipating things being worse, partially because the flu comes in. And if everyone continues to wear face masks and gets their flu vaccine, and the flu is not as severe, that makes it a lot easier in helping in figuring out which of those patients we can send home and which we need to admit. Just because the symptoms are so similar between COVID and the flu. So if you've had a flu vaccine and you're probably not having the flu symptoms, then it's more likely to be the COVID, and that makes it a little bit easier in discerning between those patients.
Right. Thank you, David. So there was a question about, I'm trying to find it. Yeah, this is a very good question. COVID-19 patients who are COVID positive, can they be shedding the virus for 2 to 3 months after the initial infection? So who would take that? Saroj, maybe I'll ask you. For 2 to 3 months, is it possible?
To be honest, I'm not certain about the data on the viability of the RNA which we detect after 2 to 3 months. We seem to have the asymptomatic persons shedding the virus, shedding the RNA, which is detected, but I don't know whether there are enough studies to see the viral, the viability of the virus, certainly not in Sri Lanka.
I think I agree with you, this is Jay. I think the South Koreans reported case series where they found 3 months, maybe about 6 weeks. And as Saroj said that, one thing to shed the virus, other one is the infectivity. So they're not infectious, but they are shedding viruses, and you can pick up on a PCR. So, but not. So the problem at this moment is that we don't have quantitative PCRs. So there are a couple of companies developing quantitative viral loads. When that's available, then you can actually give a good answer.
Yeah, that's great. Yeah. Another question is about senior citizens requiring regular flu vaccine versus high dose flu vaccine. I would say for senior citizens and everybody else, regular flu vaccine is enough, and some studies have shown that before. I don't have data with me right now, but this concept of high dose flu vaccine was introduced for people over 65 years, specifically for over 65 years, because the immunogenicity or the ability to produce antibodies is much higher with high dose. But that has been, of course, debated over the last few years. But for the most part, I think your regular flu vaccine is good. But if you're not happy, go ahead and get yourself the high dose vaccine, which is a little bit more expensive. Anything to add to that?
I think that's about it.
Yeah. And then another question: the immunity from the flu vaccine is good for 3 to 4 months. Hence, when is the best time to have the flu vaccine? I think I answered this before. The best time to get the flu vaccine on a regular year would be end of September or early October, right? Typically, the maximum activity of the flu vaccine will last for 3 to 4 months. So that will cover the flu season, right? October, November, December, January. But in this year around, I think I would take it in September, and this is September, and we're talking about, most vulnerable people who could potentially have run into problems with influenza, such as post-influenza viral pneumonias caused by influenza, and post-viral pneumonias caused by Staph aureus and other bacteria. And especially we lose about 60,000 patients every year to influenza and influenza complications every winter. This time around, we don't know what will happen, but I don't want to scare people unnecessarily. Just take the flu shot and get done with. All right, so I think we have come to an end. So thank you so much for your participation. Once again, it was a pleasure to have everybody. I was honored to have Prof. Walter Jayasinghe, one of my professors, for sure. And then Anil was part of this as well. And then Devinder and Dushanta, Malini, everybody pitched in with your knowledge. Thank you for sharing your knowledge, for sure. And this virus has profoundly impacted the entire world. So we continue to do our share of work, and we continue to share our knowledge with you guys. Next few months, we will come up with more theories, more knowledge, and then Keshini will tell you what's happening next month. We're going to put together a business talk for people who have lost their livelihoods, their jobs, and how to find new jobs, or where to find new jobs, and so on and so forth. Thank you, guys. Keshini, over to you.
Dr. Deepthi, wow, it's a lot to absorb, but excellent presentations. Thank you so much. I don't want to tell the doctors, you guys are our heroes. You're risking your lives to save others. Thank you from the bottom of my heart. And Dr. Saroj, it's almost past midnight in Sri Lanka. You're still with us. Thank you.
Yes, delighted to be with you. And thank you once again. Thank you very much, Deepthi, for your kind words. And great to have met all of you and my friend, Dushanta. Thank you very much.
Thank you.
You too, but we have our Vice President, Ayesha Jayasinghe, is going to convey the note of thanks. Ayesha, over to you.
Hi, this webinar was so interesting and so informative, and we are really thrilled that we have such experts who have proven information, and it was very enlightening, and we want to thank all of you for taking the time and effort and energy to be here and to give us a lot of information that is so important to the lay people that don't get this kind of insight. So I want to thank you all very much, because I really enjoyed the show, and I'm sure the others did too. And I want to thank the Sri Lanka Foundation staff, because you were doing excellent bringing through with the presentation and all the work that goes into making this happen. And I want to thank the listeners for coming in and hearing what is available and what new things are happening. And I think we all benefited. So I want to thank you all. Thank you.
Thank you, Ayesha. And we'll see you all at the next webinar.